Recording has started. Opening it up now.
Thank you, Tony. I'll miss you.
You are very welcome.
Yeah, he's my Iron Man, and last, roughly half a year, basically without him, I cannot do anything. But I learned a lot during this time. Oh my God. Oh, yeah, I miss him. I mean, that's basically what I'm. So we can chat. I mean, while waiting, people, people will join soon. So, how's your weather? The East Coast.
It's freezing. It's 5 degrees Fahrenheit today.
Oh my God.
Very, very cold.
Yeah, similar to Boston. I think.
Oh my. Boston is also single digit.
It's 5 degrees. So what is that? Minus 15 Celsius or something?
Oh my God. Oh my God.
So
The Boston temperature, what is that?
I think it's minus 9.5 degrees. So it's below zero already.
Okay.
Yeah. I think there will be a really heavy snow. I think this weekend.
Oh, really?
So you can go skiing.
It's a nor'easter coming, so I don't know how much snow. You guys usually end up getting the big snow when a nor'easter comes up the coast. We're probably going to be a little bit isolated because we're a little bit inland. But yeah, last year we got a lot of snows. This year a little bit, but not so much. Not so much.
Yeah, it's the same here. I mean, I saw a little bit flake in the morning, not little, but now clear up, and then now we are beyond freezing temperature right now. So
yeah.
I'm lucky in a sense.
Yeah.
I think I think Boston has a kind of warm winter this year. I think comparing with last year, it is warmer actually.
I visited November Boston.
Oh,
so
wow.
warm.
Yeah. We thought, this year is so warm, and what happened? And because last year there are several really big snow, and
Well, we'll see how it will go. The good thing is that we have to wear masks, so now wear mine outside too, so that I don't get cold. Keeping my face warm outside.
Yes. Actually, I like the mask now because that protects some heat loss, in a sense. So that's kind of good thing. I mean, in a sense. So I mean, we are adapting. So human always.
Yeah, we still have the mask mandate in the university, but my guess is that not too far from now, I think we're gonna do away with the mask mandate. I think a lot of those federal mandates are expiring, so I think we're probably gonna go back to whatever new normal is.
Right. So you're still teaching online?
No. We're still teaching it. Almost everybody's teaching in person.
Oh, really? So actually, we plan to, but we have the 2 weeks buffer period, so everything online right now, and then we will open up, start to, the in-person teaching starting, I think, the last day of the January.
No, we went to full in-person last semester in the fall. And I guess both the instructor and the students have to wear masks in the classroom. But everything is in person now. And
okay.
I mean, there's a lot of. I mean, I had 2 of my students went down with Omicron. I had to do like a Zoom plus in-person thing where I would do like the 2 modes, post my lecture, bring my camera with me and everything, and do that.
Oh, shit. I mean, I heard a lot of people, got, COVID recently.
Yeah. Omicron is crazy, and
it's incredibly contagious. Yeah, I've got 2 of my grad students also got COVID. Their girlfriend got COVID. She ended up in the hospital, actually. They say that it's mild, but I've had 2 people that I know that had pretty severe reactions.
Oh, shoot, that's not good. So she's okay now?
Yeah, she's fine now, but they had to take her to the emergency room because she couldn't breathe, and
Wow.
Oh my
yeah,
God.
they had to get a little oxygen, and then now she's fine. But yeah, you just never know. I mean, it is a nasty virus.
Oh my God. Even she vaccinated still have something.
Yeah, she was vaccinated. I don't know if she was boosted or not, but she was definitely vaccinated. But I did.
Okay.
It
Yeah, I
looks
mean,
like the yeah.
probably vaccination can help, I mean, the more severe thing, I guess.
Yeah, well, it depends if you are unlucky and you have the wrong HLA combination of genes that don't see the virus very well, so you don't express it. I mean, I can
Yeah.
see that could be, but usually it's a mild case from when you look at the statistics.
Right. Oh my God. Okay. But I glad, I mean, she's now fine. I mean, so that's I mean, thank God. I mean,
Yes, now she's okay, but it is a way. And I had some other friends here in town that they told me that it felt like the worst flu cold that they had in their lives. You know, they were like in bed for 2 days. They couldn't move. They were very tired. So it was. They told me that it was, you know, even the Omicron was pretty severe viral infection to have.
I see. I mean, you know what? I went to ER multiple times, not because the COVID, but before, some other, other my problems. So I know how, terrible or horrible in terms of going there. I mean, as patient, also, family member or, some beloved ones. So, oh my
Yeah,
God. So
you
we should
don't
end.
You don't want to go to the ER, that's for sure.
Yeah, we should end this kind of, craziness. Okay, so I guess we need to start. Okay, so thank you so much for all of you. So good morning, afternoon, evening. Hola, hello, bonjour, ni hao, konnichiwa, 안녕하세요, and hello all. And welcome and thank you for joining us the 18th seminar and Penn State Day. I can't believe it's already almost the end of January and the 18th seminar of this series. And as I said last week, almost 2 years of the pandemic have changed our views of seminars, conferences, and teaching. However, one thing does not change, and scientific discussion must go on. So that's who we are here today. And SynBYSS, the seminar series, started with several hundreds of audience for the first seminar. However, I got so many emails after the first seminar, especially from Africa, Asia, and Australia, where their time zone is not convenient for this seminar, requesting sharing of the recorded video. So in response, I got permission to release the video from all the speakers, and then I became a YouTuber unexpectedly. And as expected, the number of live audience decreased tremendously after the first video release and then follow video release. And while most audience now watch the YouTube videos, now with total of roughly 16,000 views for 17 videos. And considering the reduced number of live audience, I decided to switch from webinar format to normal Zoom. So in this format, if you wish, you can show your handsome or pretty face and ask a question by unmuting starting next week. Or if you're shy, you can put question and comment in the chat box as we normally did last half years. And today we still keeping the webinar format. And I believe we as human should adapt to the changing environment, including COVID and also climate crisis or emergency, I believe. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Costas Maranas from Penn State University. That's why I call today Penn State Day. Well, he is a math and computer science genius, and more importantly, one of my first collaborators. In fact, I met him for the first time in November 2006. Oh my God, 15 years ago, when I was just a PhD student, actually second year PhD student, after my 5.5 years of industry experience in biotechnology company. And he was, of course, a famous invited speaker, I believe, at the conference in Korea, and actually Sang-Yeom Lee organized. However, to me, his position or reputation was not that important because all I remember is we've just drank together and a lot, because Sang-Yeom Lee, the chair of the conference, provided diverse Korean free alcoholic beverage. Yes, the free beverage, a lot, unlimited. And of course, I did not expect him to be my first collaborator when I started my professor career here in 2012, 10 years ago. So I want to say, what a fate. And we are all connected in a sense. So Costas, it's my great honor to have you. And the virtual stage is all yours now.
Okay, thank you, Taesung. Can you see my screen? Okay.
Yes, and the slide board, yes.
Perfect. So yeah, thank you for the introduction. You're right. I have fond memories of that meeting in Jeju Island in South Korea. I met a lot of friends there, and I made many new ones. And I don't remember many things because, as you said, I think we were drinking too much. So yeah, today is not about me. It's about Fang Hong, who's going to give us a long lecture later on his work. So briefly, I'm going to tell you a little bit about our work, how I came up, how I ended up working in this area, some of the decisions that I made on the way, some of the lessons that I've learned, some of the good practices and bad practices to avoid over the last 20, 25 years that I've been, in academia. So here is a picture of our new building on the left. So you can see that it's a brand new building. So I know that COVID has curtailed our travel, but if you end up at Penn State for whatever reason, please don't hesitate to stop by. I'll definitely give you a tour of the campus and we'll be gracious hosts. So if the ones of you that are outside the US and you're wondering where is Penn State, is right over here. It's right in the middle of Pennsylvania. There is beautiful nature all around it. So I think that if you decide to come, I think you'll enjoy it. So if you look online and try to look at my publications and you go back in the 90s, you'll be a little bit perplexed. You're going to be seeing things like batch plan scheduling and two-stage modeling of multi-stage midterm planning. So you'd be wondering, is this the same Costas Maranas or is this a different one? And the answer, yeah, it's the same person. So my background is in chemical engineering. So when I started my academic job at Penn State, I was solving traditional chemical process systems problems. But after a while, I became tired of solving the same problems and trying to compete with other people to solve them a little tiny bit faster. The human genome sequencing effort was blossoming at the time, and I realized that all the tools that I've learned to solve process systems problems nicely apply to address challenges I have to do with either sequencing or understanding how living systems work. So despite the protestations of my academic mentors, I decided to switch areas and irreversibly move towards biology. So So essentially what I did in my career, I guess in a nutshell, is that I took all the lessons that I learned from chemical process plants, detecting errors in your flow sheet, redesigning your plan, figuring out what is in your plan, and I reinterpreted all those mathematical tools in the context of biology. So we try to figure out what's under the hood in an organism, what is the complete chemistry repertoire of an organism, try to coax that organism to do something else, and in more detail, identify enzymes and try to change those enzymes so they can do different tasks. They can recognize different substrates or operate with different cofactors. So that's kind of in a nutshell my career in biology and synthetic biology. It wasn't called synthetic biology back when I started. I remember suggesting making 5 genetic interventions, and people were looking at me as if I was crazy. But nowadays, this is an afternoon's work. So this is kind of where it all started. I remember I needed to go to the Penn State library, the paternal library, and I was looking for a paper at Chem and Science, which is a pretty nice journal in chemical engineering journal, probably some scheduling paper that I was looking for. And back in the day, you actually had to go to the library. You couldn't just download a PDF file. And what I found as I was browsing through the journal, a review article by Francis on directed evolution, and I couldn't, I just started reading it, and then I realized that, boy, this is, that's a complete new area where you can use, a protocol as a discovery engine. So we started working in this area, and I guess the future was what it was, right? So what are we up to these days? So we're working essentially in 2 areas. One is on networks and metabolism. So as I said earlier, we try to reconstruct the metabolism of different organisms spanning all kingdoms of life. We are trying to find ways to teach those organisms to do new tasks by reaching and making a new product that could make natively. We're trying to explain metabolism and try how to measure internal metabolic fluxes. And more recently, we're trying to do some kinetic modeling, introducing some mechanistic detail in all those models. And we're members of a couple of bioenergy institutes. So that helps us amplify our contributions and make the goals that we're setting out for ourselves more relevant. So this is on the pathway end of things. On the protein end of things, as I said earlier, it's the same underlying mathematical structure. We're trying to optimize over what amino acid you're going to put where in a protein so that protein will have the right functionality. And that protein could be an enzyme that you modify for a new substrate. It could be an antibody that you modify its variable region so it will recognize a new epitope. Or it could be a pore where you engineer the interior of the pore so you can do specific separations. And actually, in the context of COVID, we did some work on trying to predict the binding between the ACE2 receptor and the spike of the virus for different variants, introducing some machine learning approaches. I'm frankly jealous of all the young people that are starting their career right now because all the pieces seems to be coming in place. So you have the emergence of both computational and experimental tools, for example, or several-fold. So right now, if somebody gives you a sequence, you can come up with a structure. You can apply machine learning tools that you can inform them using both sequencing and structural features and make functional predictions. After that, there are detailed algorithms that will allow you how to predict what amino acid changes to make so that you can have a new part. And finally, as we'll hear later, we have a suite of different genetic engineering tools based on CRISPR-Cas9 and other approaches that will allow you to instantiate all your designs. So that we have we have managed to close the loop. So at this point, I think young synthetic biologists are only limited by their imagination and creativity in solving real problems, both in biotechnology and in biomedicine. So I will leave you with just a shameless ad. We're looking for a couple of postdocs in our group, both in the pathway end of things and also in the protein design end of things. So if you're interested, please contact me. But as I said earlier, today is not about me, it's about Fan Hong, and I will yield the remaining of my time to his presentation.
Thank you. My computer temporarily frozen. Says that's amazing. Wow, an amazing talk and inspiring insight with your career history and the field of history. And of course, I mean, Professor Francis Arnold was the first kick-off seminar speaker who also changed your direction, I believe. And what an interesting story. And I think, I mean, you have adapted and evolved in response to the scientific progress, I believe, and probably accept your slight background color, even though I love that color. And I used to use that, background color before. In the industry, actually, I use that, color. And then, yeah, love it. Okay. So also, even I did not provide our course today, you did wonderful job to me, to us.
Great.
Thank you so much. Thank you so much. It's brilliant talk. Okay, now finally, the main speaker of today with a little bit longer introduction. Dr. Fan Hong is a postdoc fellow of Peng In's group at SynBYSS Institute at Harvard University. As you may know, Peng In is a pioneer in synthetic biology, and his lab used synthetic DNA and RNA to construct, manipulate, and visualize nanoscale structures. So I believe, I mean, Dr. Hong basically selected to join his lab. That means that he's the true rising star. At SynBYSS, Dr. Hong is focusing on the development of programmable molecular tools to study RNA biology and control RNA gene expression in cells. And before joining Harvard, he obtained his PhD degree from Arizona State University, where he worked in computational DNA RNA design to forge complex nanostructures, RNA machines to regulate gene expression, and computational tools to study DNA RNA folding. Dr. Fan Hong is also a co-founder and founding scientist of SynBYSS, a startup company dedicated to develop home test device for COVID-19. Of course, we all need those kind of device, really desperately, and then he is kind of contributing to this society by developing those kind of important device. I'm very interested in such research, of course, and who would not be interested in such research? So we should become a fan of Fan. So Fan, take it away.
Thanks, Dr. Amoy and Maranas. And this is amazing opportunities that to share some of my research I did in my past few years.